right, I am thrilled to welcome to the stage our Friday keynote panelists, and Mike Kelly will be moderating today, so we can have them join us on stage now. Excellent. This is going to be really good. We have some wonderfully involved and smart people here to talk about the future of research. Excellent. Thank you. Thank you, Mike.
Ain't no illusion. Try to click with what you got. Taste every potion. Cause if you like yourself a lot, go let it out. Go let it in. And go let it out.
Good morning, everybody. Thank you for being here. It's nice to see all of you. It's nice to see so many of you last night. It's always a pleasure to have everybody back together again. On the stage, I'm joined by 5 key opinion leader neuromuscular scientists that most people know and have heard of. I'm not going to work my way through an introduction. I'm going to ask them to introduce themselves, their institution, who they are, and a little bit about themselves. And we're going to get on to a number of topics that we're going to discuss. So first, Carsten, would you like to introduce yourself, and Dan as we go down the line?
Oh, yeah.
Yep.
Yeah. I'm Carsten Bornemann. I'm a pediatric neurologist, neurogeneticist, and neuromuscular specialist. I work at the National Institutes of Health and an NDS in the intramural program. My history with Duchenne is long. I actually did my postdoc with Luke Kunkel in Boston, who, of course, found the dystrophin genes. I have a long history with that particular gene. Glad to be here and involved in many projects around gene therapy as well.
Hi, I'm Dan Levy. I'm a physician scientist working at Pfizer, and I lead our gene therapy program for Duchenne muscular dystrophy.
I'm Kevin Flanagan from Nationwide Children's Hospital in Columbus, Ohio. I'm the director of the Center for Gene Therapy there and lead the neuromuscular clinical program. And I'm a physician by training, but have an active laboratory program as well and clinical research work on gene therapies and gene splicing therapies, as well as gene replacement therapies.
Hi, I'm Alison McVie-Wiley. I'm from Vertex Pharmaceuticals. I landed at Vertex Pharmaceuticals through the acquisition of Exonics Therapeutics, which of course, we're very thankful to the support of Cure Duchenne for Exonics Therapeutics. And at Vertex, I lead the Duchenne muscular dystrophy disease area there. I'm a scientist by training. Before Exonics, I was at Sanofi Legacy Genzyme and have worked in muscle diseases for a number of years.
I'm a medical geneticist, and I've been in the rare disease industry now for 30 years, working on developing rare disease treatments at BioMarin earlier, and now I founded a company called Ultragenyx that works on rare and ultra-rare diseases. And part of that time, I've done a lot of work focusing on regulatory policy and the regulatory process, which we think needs substantial improvement in order to address the next generation of treating rare and ultra-rare diseases.
Thank you all. There's a number of topics that I want to discuss today, and the first is really going to involve where we are in industry. We're on the cusp of the approval of the first gene therapy product for Duchenne. I heard Deborah mention earlier on about the PDUFA date as well as the AdCom meeting that's coming up. And that's one of a number of programs. There's other companies, Pfizer themselves, should recruit out next year. And following in the heels of that will be REGENXBIO and Solid. And as we look at that, clearly I think a topic of discussion right now is one where I'm going to lean into both Carsten and Dan to lead, but the whole group is welcome to involved in this. And it's really to get a handle and an understanding on where we are today with where we are with serious adverse events and safety. I know that the community is incredibly excited about the potential for gene therapy, but I want to make sure that we're all aware of the full package that we're starting and understanding that we're building around this here. So, Carsten, I know that you've been involved in a consortium, and along with Dan and others. Could you just give us a sense of where you are and the updates on this?
So I'll start if that's okay.
think this was about a year and a half ago, maybe even a little bit more, that we saw in one of our clinical trials patients developing pretty severe weakness about 4 to 6 weeks after receiving the gene therapy. With some unusual neurologic symptoms, things seemed odd to us, and we believed that it was caused by a reaction against the miniaturized dystrophin that's being expressed in the muscle cells of the patients after they receive gene therapy. And around that time, we were contacted by our steering committee chair, whose name is Francisco Montoni, who advised us that we might need to speak to some of the other companies. Evidently, they may have had some similar kind of reactions. So we did. We reached out to the 3 other companies that had ongoing gene therapy trials. We set up very quickly a confidentiality agreement so we could all talk about safety together. I'll say that that's fairly atypical for pharmaceutical companies in general to kind of get together to talk about these things, but under the circumstances where this is really a new field for all of us, and really in the spirit of really trying to protect the safety of Duchenne patients, we got together, we shared our data, and then we went back to Dr. Montoni and also invited Dr. Bonneman to speak with us briefly, at which point we agreed to set up a panel of scientific experts to review all of this data confidentially, to try and interpret it together, and to come up with some recommendations about what we should be doing here. So with that, I'll pass it on to Karsten, because I think he kind of took the lead from there. Dr. Bonneman.
I have to say, it was a remarkable experience. Of course, it was all starting with fairly serious adverse events in these boys in these clinical trials that were really concerning, taken in their isolation. But the recognition that it was happening across a number of different trials told us that there was something bigger going on, not just a problem with one of the products that was in clinical trials, something more general that had to be addressed. And the speed and willingness with which all sponsors and the scientists, including Dr. Flanagan, came together to analyze this data almost in real time was really something I'd never seen or experienced before. And I think this is a wonderful model going forward, and hopefully we'll talk about that later. What came out of that was a very quick recognition that there was something very similar about all the boys who had these adverse events and their particular genetic makeup, their particular mutation, and how their genetic mutation influenced the reaction of the immune system against the therapy, the therapeutic transgene, the miniaturized dystrophin. And that allowed us to very quickly de-risk the trials for new incoming patients by avoiding this particular clash between the genetic mutation in the patient and the transgene, so that the immune system wouldn't be upset. We'll talk about that a little bit later more. It also allows us to then say, well, yes, so that's the immediate response to this, but of course we have to come up with a way forward so that every boy with Duchenne muscular dystrophy can, in the end, be treated. And so that set off and still ongoing collaboration, a collaborative effort to understand the individual risks, how is this best addressed, what are therapeutic avenues forward, how can everyone be included and treated in the end without risk to individual patients or undue risk to individual patients. So I think this has been very, very successful, and also I think it was fairly quick in communicating back to you guys and Duchenne and all the advocacy organizations in the space. We had a webinar, invited everyone in to just kind of transmit the data very quickly back to the community so that we are all on the same page going forward with these therapies and not being panicked or saying what's going on. A good model, I think, to address future safety risks that will emerge for sure as we go forward with this. And we can talk about that also at some point. But maybe I stop here and just to say that this is still an ongoing project. The working group is still there. It's expanding. It's expanding to address additional risks that have emerged in this space, including the thrombotic microangiopathy that you've heard about, risks that are related to the vector itself, to the gene therapy vectors that are being used, and also really drilling down to individual risks of patients and their disease. To really be able to come up with strategies to be nimble in our reaction to that, react and put safety measures into place as we go forward. And I can elaborate on that a little bit more later.
Do you think that as we get closer to approval, there's guidance that will come out that will allow patients to make better, more informed decisions about where they may be in the risk profile?
Yeah, that's, we discussed a little bit before the panel. Guidances are, of course, always a difficult thing for a sponsor to pull out of their head and without going complicated approval processes. But I think if we have a consortium, a group of sponsors, academicians, providers, experts, we can still come up with relatively quick guidances and standard operating procedures that we can disseminate and compare amongst each other to make sure that we kind of in a fairly non-bureaucratic way are reacting to anything that emerges. One has to say, I think it would be dishonest to say that this is the end of adverse events that we're going to see in this space. We're going to see more. As we go out and dose more and more patients, if this gets approved and it goes out into common usage, there will be more. There may be severe adverse events that will be emerging. That should not grind us to a halt. That should not set us back. We just have to be able to react to that, understand it very, very quickly, to then be able to put management and strategies in place to mitigate those risks, preempt them, understand the risk profile of every individual patient that goes into these therapies. This is, of course, a very new, it's a transformative type of therapy, but it's in its infancy. If you remember when the first organs were transplanted, the first heart transplantation, the first kidney transplantation, one had to learn patient by patient how to best manage that, how to best treat proactively and after treatment. And in a way, what we're doing is very similar. We are transplanting a single molecule, the dystrophin, into a patient. So we're doing some sort of molecular transplantation, and we're at the very beginning of that. We're learning at a rapid clip because we're not just learning within the Duchenne field. We're learning from everything that happens in gene therapy out there. This is a tool. It's a class of tools that we're using. And so for the effects that are related to, let's say, the AAV, to certain transgenes, we can learn from other efforts in different genetic diseases and be faster in understanding and adjusting our therapies. And then there are, of course, safety risks that are patient-level risks that we also need to understand. And we understand them better the more patients get treated. We have to be aware that we will encounter situations where certain patients are particularly high risk without having that recognized before. And then we have to understand what is it that puts that patient at risk, what is it that makes another patient a very low risk, and how can we understand that before we treat and treat proactively. That will be the strategy going forward. It only works if we talk to each other, if we compare the data, if we learn rapidly, if we don't silo ourselves off, but really compare notes in real time as we go ahead. And then we can, I think, move forward.
And do you imagine that the extra risk and time that we need through it, it'll actually impact the rollout of these sorts of products and access to patients? Do you think we'll be extra careful in trying to look for certain adverse events?
Well, let me not talk alone. Let me pass if anyone else want to.
Well, I'll speak first. I mean, I think that there was a fair bit of research in hemophilia patients on gene therapy. We learned quite a bit from those trials before any of the DMD trials were begun. And I think that with the DMD trials and with some other new gene therapies that have come along, the community overall continues to learn. And what we are sharing from these experiences are helping the next generation of gene therapies or gene therapy type treatments to move forward with.
Mm-hmm. Okay.
Yeah, I would just second that. I would say that I think the work that you guys have done in pulling together the field and different sponsors and different companies to be able to look at this more holistically is something we can all learn from, absolutely. And everything that happens, I think, in gene therapy trials, not just in Duchenne muscular dystrophy, but, Dan, as you say, in hemophilia and other diseases, we can learn from that and make sure that as we're designing the next generation of gene therapies or other types of therapeutics, that we take all of that information into account.
Yeah, it is always, it's an integrative process, learning from that, because there's things that are very similar across different gene therapy programs. Or we have a certain AAV serotype, maybe when the CRISPR comes, we have certain CRISPR proteins that are the same. But then there's always levels also that are, the individual payload, what transgene, what are you putting in there, that carries its own profile. Then there's the disease itself. Every disease has different risk profiles, and there are risks that are disease specific. If you probably know of, in the myotubular myopathy gene therapy trial, the number of boys died in liver failure. And so initially when that happened, everyone was saying, well, this is terrible because now we know gene therapy destroys the liver. No, it was not so. It was actually a disease-specific liver susceptibility that made these patients particularly prone to having this adverse event. And now understanding this can be proactively addressed again, but it's a disease-specific risk factor that comes together with the gene therapy itself. And then there are individual patient-level risk factors as well that we understand the least, so that a patient may have different thresholds of reacting to something immunologically. So one boy is fairly resistant to thrombotic microangiopathy, for instance. Another boy may be more susceptible to it. And then the answer in that may be just in the genetic makeup of individual patients getting gene therapy. So we have to understand risk at all these levels, from the class effect down to the individual patient, and integrate them to move forward safely. Again, only if we share data this will be possible.
Thank you. Let's switch gears a little bit. I want to bring Emil into the conversation because this is the 40th anniversary of the Orphan Drug Act, and we've been using accelerated approval for over 30 years at the FDA. And yet we're talking just now about genomic medicines that can actually target the root cause of diseases, particularly rare diseases. We can go in and modify DNA and play with RNA and really change the course of disease. Emil, do you feel, and I know where you're going to go on this, but do you feel that we're well set up with the way the agency runs today to think about rare and ultra-rare drug approvals using the tools that we've got?
Well, I think the Orphan Drug Act certainly opened the door to getting treatment started, but I think what's happened lately, there's certainly a lot of new technologies which give us the opportunity to do something we couldn't do before. But the FDA is still operating with a 20th century mindset in terms of what it takes to prove a drug. There's a standard of using how a patient feels, functions, or survives, but that works when you have really quickly reversible treatments to diseases with simple treatments. When you're talking about complex diseases, muscular disease, neurological diseases, it starts to get harder to design and develop drugs using that paradigm. And what I think has happened is that there are many rare and ultra-rare diseases for which that paradigm just won't work very well. The trials would have to be multi-years in length, would involve some group of patients getting harmed as part of this controlled format, and ultimately will not actually optimize the treatment. The truth is that for complex diseases, as the ones we're talking about here, you need to get the first treatment approved to begin the process of medical evolution, where the next comes and the next comes. And for complex heterogeneous disease with variable time courses, it's very hard to do. And I'll give you the best example and the really important legislation around accelerated approval. which is HIV infection. Now you might say, what does HIV have to do with Duchenne? But in HIV, there's a genetic cause. In this case, it's a virus. It comes in, but the time course of what happens to you may vary. You just talked about all these other genetic factors. But it'd take you 7 years to get into AIDS and into that problem, and it's variable between different people. Now, when the accelerated approval legislation happened, we had a first biomarker and a way to measure the disease, but people fought it. They didn't like it. But what happened is you're able to get a first drug approved. Maybe not a great drug, but a good drug, something that worked. But very quickly, another drug came and another drug came. And during a 16-year period after 1992, there were 29 HIV drugs developed, including 4 combination drugs, and a disease which we all thought was a death sentence, now a chronic managed disease. The key to that is not that the approval of each product was a little bit faster. The key is that we're actually focusing on the underlying disease. We're treating the underlying disease in a way you couldn't using opportunistic infection, hospitalizations, or immune function. You couldn't use that. You could actually address your science to the problem and know that HIV virus of 50 meant something and 0 was better. The same is true now in our rare disease. We know exactly genetically what's wrong, and we should design our treatments to deal with the thing that's wrong rather than looking what happens over many years and trying to use variable clinical endpoints that are highly impossible to do when you can do short randomized trials that don't harm anyone, allow you to prove whether this drug is doing what it needs to do, get the first one approved, and whoever that is has that approval should know they're on notice. Someone else may come after them with something better. Mm-hmm. And that is how we get to a Duchenne that's treated. And it may end up being combinations, or it may be a better version of gene therapy, but we won't get there if we create a lot of obstruction, make it very difficult for the first one, and then create a paradigm of complex trials that result in the inability for the follow-on products to occur. Now, I know that's what's happening today because I got a product approved for a disease called MPS I, a lysosomal disease. That approval was in 2003, enzyme replacement. I knew it was a good drug, but I knew it wasn't great, and I thought soon there would be improvements. But now 20 years have happened and no improvements. Why? Because they didn't let us get approved on the biomarker. They made us use complex clinical endpoints. The improvement drugs have a difficult time running that kind of a trial in patients with complex clinical endpoints. They have to run very large trials, and therefore they're obstructed by the very process. If they had focused on measures of the underlying disease, the source of disease, we would have iterated several improvements. So right now on deck with the Sarepta situation, accelerated approval, is really the future of Duchenne and other neuromuscular disorders. If we're not going to get our heads around how to approve something on a biomarker, even not a perfect therapy, a therapy that's the beginning, we won't get into a process of medical evolution where we'll obsolete that with a better one and a better one and find ourselves instead in 10, 20 years from now with a really great Duchenne treatment. And so there's a lot at stake with this situation, and I've been spending this year going after the FDA, and I had a meeting with the commissioner and all the center directors, and I challenged them with the fact that what they're doing is not scientifically sound and not right, and they need to start using accelerated approval pathway for our rare and ultra-rare diseases, and that failing to do so will result in the loss of many treatments, not just for Duchenne, but I'm talking about for other diseases which are on the brink right now, for which doing a multi-year randomized trial is impossible and unethical. And so there is something very important at stake for all of you with regard to getting accelerated approval for the first one. However good or bad you think the treatment is, the first one opens a door to improvements going forward.
Yeah. Thank you for that. I knew exactly where you were going to go. I've heard you talk about it a number of times. I wrote a couple of things down. There's some of the most awful words in the language of accelerated approval for me were reasonably likely, and trying to figure out what that actually meant. And so we really need to figure ways in order to improve this process. I think it's it's a demand on patients really to go into placebo-controlled trials, particularly in diseases like this that are unrelenting. And I think the final piece that I'd add in there really is I don't know how we're going to effectively do combination treatment options in this disease that we know are necessary and coming without the agency actually allowing for different ways to regulate drugs or approve drugs.
How are we going to do it? Combinations? Well, I think while we focus in drug development on a single endpoint, truth is, when you go to the doctor and you see the doctor, do you just talk about one thing? Is that how you measure your health? Never is. Yet, the statistical world has driven us into this simplistic thinking. So when you talk about combination therapies, you need to talk about combinations of evaluations to understand the benefit or risk of what's going on, right? And there may be one biomarker, how much microdystrophin or dystrophin you have. There's another ones may relate to muscle integrity or other things. So we need to get a little more sophisticated in building in our analysis across the whole spectrum of the science that we have and get out of the statistically driven idea of a single thing as the only way you make a decision, right? And that's, I would say, core to figuring out combination therapies is being able to look at multiple things that are happening and be able then to triangulate whether you're showing an improvement or not. So that to me is fundamental to us moving the next level. And in HIV, that did happen. They started with a CD4 count, then they went with a viral load, and they got more understanding of how much viral load reduction. And then it turns out using both of those is even better, the combination of the two. And so over time, they started evolving the understanding. I think we need to do that in Duchenne as well. Start with this first one, but then start building in the others, understanding how they relate to each other and how they describe. You know, and what I can say, I always tell people, think about Star Trek and the little tricorder device. They figure out how well you're doing, right? Well, the tricorder doesn't do the 6-minute walk test. It doesn't do that, right? It's measuring several different biomarkers that it can detect somehow, and it's putting them together and synthesizing them. That's kind of the direction we want to go.
Okay, thank you.
It's not going to be the 6-minute walk test.
Yeah. Yeah. You know, we spend so much time, effort, and focus really trying to de-risk programs or accelerate research, but quite often it's really what you're describing, you know, getting things through the FDA and actually approved and onto the market is quite often the major bottleneck that exists in there, and how we work through all of that is going to be, has to change over the future. And I appreciate what you do. Thank you for that. I'm going to change topics yet again, and really, we've talked about some SAEs so that the audience will know where we're going to be in a few months' time whenever the first gene therapy product's approved, the regulatory side of this. But the future is really one where we're looking to see some of these next generation of therapies come through. And we've got two individuals today that could talk to some of that. I think from the work that Vertex has done in cystic fibrosis leans heavily into where we're going with Duchenne and really understanding what a cure may actually look like and what patients could expect. Or the work that you're doing with CRISPR-Cas on the exon 51 amenable skipple patients, you know, really speaks to what a cure may actually look like for those patients. A single treatment with a rewrite on the front of the exon. And Kevin's work recently with the DUP2 patients, and particularly the young man that's been treated recently, you know, I think really speaks to some of where we may be in the future with as these therapies come through. So I'll invite the both of you, you know, just to make some comments and some guidance for the audience about what to expect.
Sure, happy to. So, I mean, I think I would say it's actually building a little bit on what Emil was saying about continuing to innovate and continuing to iterate on treatments as new technologies come along that potentially have application to a disease like Duchenne muscular dystrophy. And as Mike said, you know, at Vertex, we built a franchise for cystic fibrosis by really thinking about that strategy. So Vertex as a company has worked on and invested in cystic fibrosis for over 20 years. And it took some time for Vertex to get to the first medicine for cystic fibrosis. That came in 2012. And that only treated a small subset of cystic fibrosis patients. But with continued innovation and with continued commitment, Vertex now has 4 medicines on the market for cystic fibrosis that has the potential to treat up to 90% of cystic fibrosis patients. And with that as well, I think it's thinking about the totality of the disease and every patient, every mutation, every age of patient, every stage of the disease progression, and making sure that we think about developing treatments, potentially different treatment approaches for different subsets of patients, but also making sure that all patients have access to those medicines, which is another thing that we can do as we think about it in the pharmaceutical industry. I think as well, it's all about, and again, it's building off, I think, some of the other discussions in the room, as we think about some of these new technologies and having a deep, deep understanding of the disease itself. And again, all aspects of the disease, whether it's skeletal muscle, the effect on ambulation, cardiac muscle, the diaphragm, and thinking about ways in which we can develop treatments for that. And then applying some of these newer technologies. So as you'll hear later in this morning in the next session, Vertex is exploring the use of gene editing, a new technology using CRISPR-Cas9 to potentially target initially patients with a subset of mutations that we think CRISPR-Cas9 can be designed to correct those mutations at the source, so basically at the level of the DNA, and really tackling the root cause of the disease. So, and we absolutely build upon the work that others have done in the field. There's a lot of commonalities. And then just finally tying some of this back actually to the discussion with Emil earlier on the regulatory side of things, we are really pushing regulators as well to understand that these learnings become applicable to what we're doing because of the commonalities, and to see what we can then do to accelerate the development of these next generation treatments. So for example, thinking about platform approaches, if we're using the same delivery system, then let's leverage everything that we've learned from the safety and the efficacy side of things for that delivery system, and we may just be delivering a different cargo. For different mutations, as we think about gene editing and potentially get into that very personalized medicine side of things, then let's leverage what we do for the first group of patients so that we can do it faster for the next set of patients and even faster for the next set of patients. And not only do that from a company perspective, but bring the regulators along with us. So there's really a lot to think of as we think about what's next. Because the field, the science is changing so quickly. It's incredible to think that CRISPR-Cas9 was only discovered 10 years ago. This is the 10-year anniversary of that. And we're already talking about, you know, treatments using CRISPR-Cas9 and gene editing. At Vertex, we have a gene editing program for sickle cell disease and beta thalassemia that we've actually just submitted our biologics licensing application to the FDA to potentially get that approved. And that's incredible for a technology that within 10 years of it being identified, that is the potential for treatments to be on the market using that technology. So we all want to stay ahead of that.
Kevin?
Right. So, well, I think this has been a great discussion already, and if I just add a couple comments maybe related to what's next, and also decision-making for parents that I think is part of the challenge for this. You know, what will constitute a cure is defined differently by people, including parents. I think many times I hear parents say to me, well, if I could stop where we are now, I would consider that, that we've got it. We've done something meaningful. That's the cure I'm after. And other times we say, well, the disease modification is great. Can we get to the root of it? Can we do better than that? That's really the challenge we're going to face. I think the short-term challenge for a lot of parents, at least in my experience, will be the recognition that, you know, not all versions of dystrophin that we replace are going to have the same function. And we know this from patients. We know it when we look at big groups of patients with Becker muscular dystrophy, various versions of miniaturized dystrophin, if you will, in Becker muscular dystrophy. They don't all function the same. And putting in levels of 40 or 50%, whatever the number is, of something that we've never observed in humans, we anticipate that's going to be good and hope it's going to be good. But there may be better choices for individual parents, and that's the challenge I think they're going to face. We know there are a lot of boys who have mutations where they, if we can alter, for example, splicing, which I'll talk about, which I have a fondness for, we can restore in many of those boys full-length dystrophin. And many of the exons, the problems with exon 51 skipping to date are just about efficiency. We know that certain exon 51 outcomes are great. We know 50-year-old people with 49 to 51 deletions who have no real symptoms, for example. So in those patients, if you could get it by a very efficient process, that's a better outcome than jumping at necessarily the first thing that comes along. So parents are going to face a decision-making process, perhaps as much as a month from now, saying, should we jump in at the first therapy available, which might be the right choice, but that choice will depend on knowing the full range of outcomes, the full range of risks, and what else is realistically coming down the pike. I think there are a lot of great things coming down the pike, which is terrific. That's great, including things to improve the efficiency for exon skipping therapies in general by targeting, by alternate methods of targeting antisense oligonucleotides to get them into muscle. I think, but I often warn patients, I often talk to parents and say, if your boy somehow looks different, there's something special about his mechanism of mutation, we should take a breath a moment. Let's look at all of the data together and make some decisions at that point about what the next step for therapy, realistic therapy will be. Combination therapies are not off the table. And if there's no reason with a successful microdystrophin treatment, there's no reason that 5 years later a boy couldn't have a non-viral exon skipping therapy for some certain mutations. It wouldn't affect the transgene function, the splicing. There's no splicing of the transgene, for example. So these kind of things are realistic potential outcomes as we move down the road. We should be so lucky to have so many efficient choices to do at one time. But I think it's going to be a complicated time for a lot of parents in the coming months as we determine the full safety, as we see the full safety and efficacy profile, and determine what insurance companies are likely to pay for as well. Are they likely to pay for alternate choices for therapies? I do think the other thing that would be critical for this is when we have therapies that work, the idea of getting the newborn screening in a way that actually allows us to push down the age of therapies in meaningful ways. We'll present data later today on an infant we've treated who had a differential response for a 7-month treatment compared to 9-year-old boys treated with a viral-based exon skipping therapy. And it just makes a strong argument that we really are going to have to identify kids at a younger age to make probably significant impacts on ameliorating the disease symptoms. So it's a challenging but exciting time. I can't quite define cure, and maybe somebody else can, but I think every family views it a little differently for us, for themselves and among all of us.
Yeah, I think that's where we're going. Newborn screening and treatment at diagnosis is certainly going to be where we end up in this. That's where the most effective treatments are likely to be. And I think that initiative has taken off, where it didn't exist 5 or 10 years ago simply because the therapies weren't there. Like today, we've got ways of having intervention, so now we need to identify the kids very, very early indeed.
No, I think that's right, yeah.
It's a good example that, when every advance that you make or anticipate brings with it a new set of questions and challenges that you have to answer at the same time. And you have to do that anticipating them, not waiting for them to happen. And so if we do newborn screening, then of course we have to have therapies at the ready that we can actually offer to patients who are diagnosed by the newborn screening. And if we treat early on, if we treat a baby pre-symptomatically even, we have to anticipate a lifetime of treatment. So we can't just say we're going to treat the baby, and then if the baby grows into a boy, into a young man, and the therapy is no longer effective because it gets diluted out by the growing body, by the growing muscle, we have to have something at the ready to be able to treat that patient a lifetime, not only at birth, but to be ready to maintain that treatment. And we're not there yet, but knowing the challenge puts everyone into high gear to try to address that challenge proactively. Even before we treat patients that young, we need to think about the consequences of that for the future.
There is a really important reason to treat early, which is to preserve the muscle stem cells. That bank of cells is being used up, and treating early to protect them
right, but there you have
pushes you down the road. Then you'll have to have another step perhaps, but the loss of the stem cells that you don't see but is happening, I think is really important.
Right, it's a great example that kind of just thinking about that brings up the challenge. We need to treat the stem cells. And our AAV vectors are not good at that at all, getting into the stem cells. And so you're absolutely right. If we want to have that durability of the effect, we need to be better and get better at it. And there's good advances being made along that route. But you need to recognize the challenge early on and then address it. I think that's a good example for that.
Any other comments? We've got a couple of minutes.
I had something I probably could say a little about the FDA, and I had a meeting with the senior leadership of FDA and to talk about the need for accelerated approval, and I came away with the fact that they still have three important misconceptions about what's going on. I think they're important to all of you here. The first thing is, of course, they see biomarkers as a compromised measure of what's going on in disease. They haven't really figured out that the clinical endpoints are very noisy and downstream. They lead you to other types of treatments rather than treating the underlying cause. So that misconception about the meaningfulness of biomarkers and how predictive they can be, or how important, in fact, to focus on them when they measure the underlying disease. I think the second thing was that they have this idea that a randomized control trial in which the placebo group gets only a little bit of injury is okay. And I don't know what parent out here would say, no, it's okay. And they actually brought up the example with disease when we're working on, Sanfilippo, that if the kids in the control group got 20% brain damage, they could then exit the placebo arm and get treated. Well, there's a whole lot of mistakes in that one. First of all, the concept that people want to get in a trial will accept 20% brain damage as the outcome for their kid. The second, of course, is the idea that you can measure it that accurately. The third thing is that in that disease, once you're in the downhill slope, you've actually missed your window. And as Duchenne parents, there's a window too. So once they get over 2 years of age, which might have happened, you find the score, they're actually on their downward slope. You have no idea whether you're going to save them at all. But that's one of the ones that I think we need to fix, because I think there's situations where we can do those kind of trials, but those situations where we need to use better science at figuring out the answer, that we don't have to sacrifice people's children to get a scientific answer. Let's be better scientists. The last really important misconception we've been talking a little about here is the fact that they really think if they approve a first drug that nothing better will come. And that's just wrong. There's been so many examples where the first treatment does bring on new treatments, but they need to change how they approve these drugs. And I think that idea of medical evolution is really important, and the most important one for us right now with this decision coming up, which is whatever you think, the first one approved will enable more. Whatever you think of the original exon skipping approval, it enabled more. And we just need to get our science straight and understand the importance of medical evolution. So those are the three misconceptions that I'm going to be working on, and if there's people interested in supporting that effort to push back and educate FDA, I'm interested in having all the support we can get.
Excellent. Thank you. We're down to the last minute.
Kevin has some.
Oh, please.
No, that's what I was just going to say. And I'm supportive. Count me in. But the challenge, of course, with dystrophin
Count us all in, I think.
Problem is, as a biomarker, as everyone is well aware, it is the biomarker for dystrophy, but microdystrophin or any sort of altered, we really can't say how much is enough when we're looking at all these other forms. So I do encourage parents who have the opportunity, who are offered to take part in studies to try to clarify that. You know, there's a rationale for the use of muscle biopsy still in the diagnosis and decision-making process for parents. And I've had this conversation multiple times, so I'm a bit of a broken record, but if a child looks very different and you're making a decision about going into a trial with some significant risk, it's not unreasonable to consider a muscle biopsy to determine how much do you have in place and look at your son and say, that form of dystrophin at this level gives us this function. That's how we learn these things. That's how we're going to advance the decision to help the biomarker argument heavily with the FDA.
Thanks for that, Kevin. We're right on time, and I just want to wrap this up with a couple of things. One is, the session this morning to me was really important, you know, to kick this off, you know, really digging deep into key opinion leaders who are intimately involved in this disease and other neuromuscular disorders, really sets the stage for what you're going to hear over the next day and 2 days. But I think for me, the take-home message of why I wanted these people on the panel was really to understand that drug development, drug discovery is not siloed. It's not embedded in any one company. You can see these organizations working together, the NIH, Pfizer, Vertex, academia, etc. It takes all of that there in order to make this much more effective, much more efficient. And the work that Emil's doing at the far end of this is critically important, trying to change the paradigm of how we get drugs approved. So I've really enjoyed this, and it was the reason to bring it together for the audience, where they can see it's not just one group, one company. There's a whole field out here that are working together to change the paradigm and make these things faster and better. So with that, please join me in thanking each of these ones for their time. Thank you.